Okay, I hope you've had time to do your bell work because we're moving right along, okay? This is the last set of notes, last information you have to know before the midterm coming up. And the test on this material is going to be Monday, a week, next week, this coming Monday, okay? Which is the, 12th, 13th, 15th, I think? Yeah. And then the rest of the week, that week before exams, we're gonna review, all right? So the answer obviously is interphase. That's when DNA replication occurs. So what are nucleic acids? This should be mostly a review, this part. Remember, you have monomers, building blocks, okay? And they are called nucleotides. And together, they make DNA and RNA. You have ribonucleic, which is RNA, and deoxyribonucleic, which is DNA. So if you just take the deoxy off, you have ribonucleic. So there's a minor difference in their molecular structure, okay? The building blocks, remember, for nucleic acids are called nucleotides. So chromosomes, again, another review, made up of very long strands of DNA. Chromosomes are found in a cell's what? Yes, the nucleus. All right, and they determine the characteristics of organisms and control what the cells do. It's the brain of the cell, remember that. And the DNA is the blueprint that the brain uses to read. And they make proteins based upon what your genes are. The genes are on the chromosomes. And you have more than one gene on a chromosome. You have multiple genes, okay? And all along this section. And they code for proteins that make your body do certain things. Even your eye color, your hair color, your skin color, how tall you are, whether you're a boy or a girl, so on and so forth. RNA is, on the other hand, is found in the cytoplasm, not in the nucleus. And it acts as a messenger because DNA is too fat to fit through the little nuclear pores, the little holes in the nuclear membrane. So the messenger RNA is thin enough, it can fit through those holes. So we're going to save the test yourselves for homework. And you'll notice that it says in very capital letters, and bold, homework. So when your homework tonight is to go home and do this, okay? So what is DNA made up of? Of what is DNA made? So the diagram below represents a nucleotide with the three parts, the sugar, the phosphate group, and the nitrogenous base. So your job is to just label those. You should have known that this was the sugar because sugars have that either hexagonal or pentagonal shape in their chemical molecular formula, molecular structure, okay? So you're going to see chains of this in a circular, twisted ladder all the way down, connected in the middle by hydrogen bonds. We talked about that in a previous unit. So the phosphate is the circle, the sugar is the pentagon, and the nitrogenous base is the rectangle, okay? So nitrogenous bases contain the element nitrogen. Hello, nitrogenous, okay? And there are 4 types of bases. You can see the different colors here. There's green, orange, yellow, and purple, each corresponding to a different nitrogenous base. And the bases are paired together based upon their shape. So these, we're going to learn about what they are in a moment, but these two, you see how now the shapes aren't exactly like that in reality, but on diagrams it's much easier to show this way, okay? And you're going to remember this. So, yeah, anyway, I should be able to draw, but I can't. Anyway, right there. What just happened? I went backwards. Yeah, anyway. Long story short, you're writing these on that little section that says, has a little arrow on it. Adenine goes to thymine. Cytosine goes with guanine. Those are the pairs. So C and G, G and C, G and C, C and G, and then T and A and T. So they always pair. Adenine will never pair with cytosine. Thymine will never pair with guanine. Okay? And the way you can remember it, always take care going. You can come up with your own words that would help you remember the order, okay? But always take care going is what I came up with, and it works, okay? So A and T, C and G. You could also do like C and G are like shaped like this, so you could remember them that way as well, that they go together, not with A and T. But, all right, what's the shape of DNA? We know it's a ladder shape, just like a ladder with rungs and handles. But you twist that ladder and you get that. That's called a double helix. Double helix. It's double-stranded, has two strands, like I said before, that are held together by weak hydrogen bonds in the center. So in here, the two strands are held together by hydrogen bonds. Now the handrails, these parts right there, are made up of the phosphate and sugars, while the rungs, these part in the middle, are made out of the bases. So here are the rungs, the phosphate and the ribose sugar, okay? And then here are the bases. Adenine, thymine, cytosine, and guanine, okay? Or cytosine and guanine. And then you take this and you twist it going up, up and up and up, or down, and you get that DNA double helix twisted ladder shape, okay? So in 1953, two scientists, Watson and Crick, shown here, figured out how DNA was put together. And then Maurice Wilkins and Rosalind Franklin photographed the DNA molecule for the first time. Watson and Crick. Might be a question on your midterm or final, okay? The shape of DNA is double helix. This is just a review. We already said that. These are just different ways to represent them. Your extra credit for this week is to create a model using the instructions I gave you for this week in week 7 in Google Classroom, okay? So there is extra credit, and it was due in class on Friday. So how is RNA different from DNA? Well, we already said that DNA is deoxyribonucleic acid, and you just take the deoxy off, and you have ribonucleic acid, okay? It's like it, but it's also made out of, in that it's also made out of repeating units called nucleotides. But RNA is different in 3 ways. It's single-stranded, not double-stranded. That's very important. Remember I told you that DNA is too fat to get through? RNA is not. Instead of thymine, instead of AT, it's going to be AU, uracil. So thymine for RNA doesn't exist, okay? And it contains ribose sugar instead of deoxyribose sugar. So here's DNA on its side, and here's RNA. Single-stranded, uracil, not thymine, no T's, only U's, okay? And ribose sugar, not deoxyribose. So those are the main 3 differences. And there are 3 types of RNA. There's messenger RNA, ribosomal RNA, and transfer RNA. mRNA, we call it rRNA, and tRNA, and they all have different uses, different purposes in creating proteins for your body. All your cells are doing this. The only ones that are not are, well, no, all of them are. Yeah, well, okay, take that back. Red blood cells can't do it because they don't have a nucleus. They don't live very long, so they just have, they're like a trailer on a tractor trailer. Okay, the brain is on a different part. The brain is different. The trailer, they just move stuff through the blood. That's all, their only job. So they don't live very long. So they don't have brains. So the messenger RNA carries genetic information out of the nucleus. Told you RNA has to be skinny enough. The DNA is too fat. So the RNA comes through, it carries the message. Carries the message of the DNA out. It's almost like, here's your computer, okay? And you want to send a file to somebody, somewhere else. You can make a copy of it on a thumb drive, right? A memory stick, and you can send that with somebody outside of the classroom instead of carrying your whole computer. You see, so it's just making a copy of the DNA so it can fit through, and then the ribosomal RNA helps form the ribosomes, and then the transfer RNA transports amino acids to the ribosome to make proteins. Remember what the ribosomes do in our organelle part of the notes, part of the class? We learned that ribosomes make proteins, but they can't do it by themselves. They have to have directions. Otherwise, they'd make everything just random, and you'd be like this. You wouldn't even, organisms would not exist properly. You need to make the right proteins. So what tells the ribosomes to make the right proteins are the DNA through this process. So again, this is just showing the difference between DNA and RNA. The fact that RNA has uracil and not thymine. So it's AU and CG, and AT and CG, okay? Next, all right, now go off my screen. Okay. All right, test yourself. That's going to be done for homework. I gave you the last one on that sheet, though. So what else has DNA? Well, obviously everything that's alive. That really sums up the entire slide. So universal. Well, we don't know if it exists anywhere outside of our planet, but universal as far as all life on Earth, okay? All living things contain DNA, and the bases A, T, C, and G. It's the sequence of those bases that makes all of these species unique. Trees, bacteria, dolphins, bees, mushrooms, and everything in between. So how does RNA replicate? There are 3 steps. 3 steps how RNA gets the information from DNA. So DNA must unwind and unzip. You've got this double helix, but there's no way to get in there. So it has to unwind, not the entire strand, but pieces of it at a time, okay? Unwinds and unzips. So imagine that the 2 rungs, the 2 handles of the ladder with the rungs, imagine those coming apart. Because the individual strands have to be read and made into the messenger RNA, okay? So temporary breaking those bonds. So here's the unzipping. An enzyme does that. So you're going from this to this and then unzipping. Like a zipper. And it serves as the template. Step 2, new DNA strands are made by using free-floating nucleotides that are complementary to those on the template strand. And then finally, the DNA is proofread for errors. So there are 2 exact copies at the end. And this is just an animation of all those, the red and the blue and the green and yellow. Those are all proteins that are doing the job of the unzipping and the replicating. So you can see it's unzipping it here, and then see the proteins come in and they make new copies. It's kind of confusing, but we're going to see a video on it. So why is replication important? So in order for an organism to grow from a baby, a seed, a zygote, a fertilized egg, all those things, it must make more cells through mitosis. Our first slide, our Bellwork slide, going back to that first question. Growth and repair. In order to make a new cell, the first step is DNA replication, right? So these new cells need an exact copy of DNA in order to function. And remember that occurs during interphase, before cell division. The double helix structure is essential because it allows both strands to be copied at the same time. That's pretty amazing. Both strands to be copied at the same time. That's why replication is important. So another test yourself, you're going to do that one for homework, right? And you're going to label those. Several slides here. Oh, I'm going to talk about this one. Okay. So this part of the test yourself on slide 22, again, this is all in your notes, but you're going to go back and you're going to look at the complementary base pairs. So A, we know it goes with T, T and A, C and G. And so these little portions are called codons. We're going to talk more about those in the second part of the notes, but all you're doing is imagining that you're making a copy of the DNA. So you're making a complementary strand of DNA. So you're matching them up, okay? Matching them up with their base pairs. All right, so how are the proteins made to do this process? Well, there are several steps, okay? And there's 5 of them right here. This is 5 of them. You have to transcribe. That means essentially copy, make a copy using the messenger RNA. Then the RNA processes it. It leaves the nucleus. You'd see it leaving the nucleus through a pore. Then the amino acids, remember those are the building blocks of proteins, they're activated, and then they are, this message tells the ribosome what order to put the amino acids in. So the DNA is copied, transcribed here, unzipped and copied, right? Unwinded, unzipped, and copied. The messenger RNA carries that message out for what proteins to make, and then the ribosome is kind of like putting the amino acids together in the right order, in the right sequence to make the proteins for the cell to do its job. So we're going to watch a video now, 5-minute video.
DNA is very long and cumbersome. The strands of DNA also code for many different proteins. To get the code for the desired protein from the nucleus to the ribosomes in the cytoplasm, a copy of the small necessary section of DNA will be made. The copying process is called transcription, and it occurs in the nucleus. It's the first step in protein synthesis, the process of making protein. The second step is called translation. A portion of the DNA is unzipped so that the mRNA can be made from the DNA like a template. That's transcription. Nucleotides of RNA match with one strand of DNA and make mRNA. RNA polymerase unzips the DNA and puts RNA nucleotides into the right place. C and G nucleotides match up, A and T nucleotides match up, but anytime the RNA places a complement to an A on the DNA, it places a U instead of a T in the RNA, Remember, RNA uses uracil instead of thymine. So, if the DNA strand looked like this, the complementary mRNA strand would be the complement, but each time there would normally be a thymine, there's a uracil in its place. The mRNA also doesn't copy the entire strand of DNA. It only copies the portion of DNA that codes for the protein it wants at the time. In fact, most of DNA doesn't code for anything. Only about 2% of it codes for protein. So this small section of coding DNA has a start and stop signal. After the mRNA is formed, it moves out of the nucleus through a nuclear pore and goes into the cytoplasm. Remember, there are 3 different types of RNA. The purpose of the mRNA is to get the protein code out of the nucleus without pulling the DNA out of the nucleus. Then the mRNA will need to find a ribosome in the cytoplasm. Ribosomes can be found on the rough ER or floating in the cytosol. mRNA is read 3 bases at a time, and these 3 bases are called codons. Now, the ribosomes are made of protein and RNA, and the ribosomes are the protein-making machines that read the mRNA code and add the correct amino acid using tRNA. Remember, tRNA stands for transfer RNA, and that's because its purpose is to transfer the right amino acid to the ribosome to build the protein. The tRNA has an anticodon on one end, which will match a specific codon on the mRNA, and has a specific amino acid on the other end. Together, these 3 parts will make translation happen. Translation occurs in the cytoplasm of the cell, wherever there's a ribosome. To get translation started, mRNA attaches to a ribosome, and a start codon must be read. It's usually AUG, and I remember that school starts in August, and that helps me remember AUG. The first amino acid is brought in by tRNA. The anticodon on tRNA matches up to the codon on mRNA. Then the next tRNA molecule moves in and matches up with the mRNA codon. This time, the amino acids form a peptide bond and link together. Then the first tRNA can detach and the mRNA shifts through like ticker tape, and the next tRNA molecule can come in. The protein grows until a stop codon is reached. Once the stop codon is read, the protein is formed and ready to finish folding to become functional. And that's the end of protein synthesis. Now, we can figure out the sequence of amino acids using the mRNA and amino acid chart like this one. Let's use an example. Starting with the first 3 letters, AUG, we can find the amino acids. The first letter is A, which narrows it down to this row. The second letter is U, which narrows it down to this column. And the third letter is G, which means that this is methionine, which is the starting amino acid. Next is CCC, which is the second row, second column, and second row on the right. But we don't even have to look at the third letter in this case, because every third letter will bring the same amino acid, proline. Next is GGC, fourth row, fourth column, second row on the right. That gives us glycine. And lastly is UAA, row 1, column 3, row 3, and that gives us stop. There's no amino acid here. It just means that the protein will detach, and it marks the end of protein synthesis. Thanks for watching this episode of Teachers.
All right. Next. Okay, so a little review here is going to be what amino acids are called. We call amino acids the building blocks of proteins. And so they can be linked together in varying sequences to form a vast variety of proteins. Chains of amino acids are called polypeptides. And so the different codons code for different amino acids. And the transfer RNA, like in that video, comes in and it has one of these amino acids building blocks on it. And the code, they have an anticodon that matches up with the codon and then binds the amino acids together to make a chain of amino acids called a protein. So like I said, the set of 3 bases is called a codon. And it's a code, right? So 1, 2, 3, and so we're gonna, and I said codons code for specific amino acids, and there are also start and stop codons. So to start off, they gave you an example in the video, but use this chart, and there's another chart in your notes as well, which we're going to talk about momentarily, but use this chart to find the amino acid for the codon above. So you're going to go to G and C, okay, and any of them code for the same thing, which is alanine. Okay, so alanine is going to be the amino acid that codon is a signal for, okay? And only the transfer RNA that can meet up with these shapes will have alanine on it. So like a lock and key. So let's practice some more. So we're going to use the same tool, and we'll use that next tool that you see in your notes, that circular one, for the next couple of practices. So CAA, C, A, and then A is here. So that's going to be glutamine, okay? And then UGA, UGA, that's a stop codon. So that's going to tell the transcription in the nucleus, translation for the proteins outside. It's going to tell translation to stop. Okay, now here's a different tool. It's very easy to use, just like the last one, so let's practice it. This is probably going to be on your test on Monday and your midterm, possibly on your final, okay, your EOC, something like that. So UCC. So find uracil, find C, so that's serine, GUA, valine. Anything with ene, obviously, is an amino acid, okay? And AAA, lysine. Simple, like a little code. Okay, so your characteristics, again, like talked about it, your sex, your height, your color of skin, eye, hair, how many fingers you have, how many toes you have, how many arms and legs you have, whether your head is up here or connected elsewhere, where everything is, whether you have hair or not, all your characteristics, where your stomach is and your heart and everything about you is controlled by those genes that are on the chromosomes. Okay, that when you get half of them from mom and half of them from dad, okay? But sometimes an error occurs in the DNA transcription, okay? In transcribing the DNA in the nucleus, sometimes a mistake happens and you get something called a mutation. Now, not all mutations are bad, okay? People think mutation, it's mutated, kill it or whatever. It got exposed to nuclear medicine or whatever, and it's mutating, like the Hulk or something, okay? Or X-Men, the mutants. Not all mutations are bad. Some mutations allow an offspring to survive. Like some of us might be carrying a mutation in our genes that makes us immune to the COVID virus. That's a good mutation, okay? So don't just think mutation's bad. So there are 5 different kinds of gene mutations. We're going to talk about each one of them. So the first one is called a missense, meaning something got mixed up in the base pairs, okay? And so you end up with a different amino acid as the resulting protein. You've got these codes that code for proteins. If you make one mistake in one of the letters, you're going to make a new amino acid, which is going to make a different kind of protein. And that's called missense. The next one's called nonsense substitution. So now, non meaning like no, okay? A codon makes it stop instead of continuing. So if you have one of these out of place, these base pairs out of place, and it creates a stop message, for the transfer RNA, it will not produce any more of that chain, and you'll get a different result, okay? So one is just replacing one to create a new missense, is creating a new amino acid. Nonsense is stopping it. Stop this nonsense. That's a good way to remember that one. Stop this nonsense. Then there are three others, and they're just shown here in pictures. So the first one is deletion. A deletion frame shift is when it mistakenly leaves out a part. Somehow a codon didn't code and it just leaves it out entirely. Insertion is when you get an extra piece inserted in the DNA. So deletion, insertion, that's pretty simple. Frame shift just means that it shifts the frame of the DNA molecule. And then finally, duplication. Sometimes a gene or a section of genes will get duplicated on the chromosome. So it'd get like copied twice or something like that. So now we're on slide 32, we're going to talk about karyotypes. Karyotypes are kind of maps that you can create for any living organism. This is a human's karyotype that shows all of the sister chromatids and their matching chromosome, one from mom, one from dad, okay? So they're all paired up. One from mom, dad, mom, dad, mom, dad, so on and so forth. Okay, so what this chart's called a karyotype, and we discussed that a while ago, but I told you we were going to be visiting it again. We're also going to be looking at them again next semester when we talk more about genetics, okay? So how many pairs of chromosomes do you see here? Count them up. 3 and 2. How many do you have? 23, right? So to make a total of 46 for human beings. Based upon this information, the X and Y, however, what gender is this? What sex is this individual? They have an X and a Y. If you said male, sorry. Funny, Mr. Keene. Male, that's correct, X and Y, okay? And therefore this one's female, X and X. But this one is also male, human, 46 chromosomes, 23 pairs, but there's something unique going on here. How many copies of number 21 do they have? 3 instead of 2. And this is called trisomy 21. And there's a question about this in your notes. A person who is born with Down syndrome has trisomy 21 is the other name for that genetic disorder. And that's when they get a copy of that chromosome. Sometimes the copying of other chromosomes, the individual offspring would never live at all. But with Down syndrome, trisomy 21 babies, the genes on here aren't as necessary for, they won't kill you if they're multiplying, okay? So that's why oftentimes Down syndrome children live to adulthood because the genes that are coded on those chromosomes, if they get messed up, they're not going to kill you. They'll make you be a different kind of person, but so now we're going to do this little background information about color blindness, and you're going to take a little test, a color blindness test. All of you in here, when you look at the screen, you're going to answer some questions to see if you are indeed colorblind. Now, most likely, if you are colorblind, you probably know already, especially around the holidays when you look at all the lights up. For example, my sister-in-law, she was wondering why everybody in the neighborhood had Barney colored lights, purple and green lights. Everyone's lights are purple and green, what's going on? And we're like, what are you talking about? There's purple, green, and red, and all different colors. She she was colorblind. And she didn't realize that she was missing out on some of the other colors that were in, especially the new LED lights. Okay, so long story short, there's this island called Pinglap in Micronesia, okay, which is like Southeast Asia, okay, and it got devastated by a typhoon in 1775. Big storm. Only 20 people survived, and they were the ones that repopulated the island. One man of the 20 people, however, was a carrier for a genetic mutation that caused color blindness, right? So they intermarried, and eventually many of them have color blindness now. And it produced phenylalanine in the place of serine. So those two amino acids. So what kind of mutation is that? When you get a different kind of amino acid being formed. If you look back at your notes, which one was that? It was the first one, right? The missense mutation. So apparently that mutation, that genetic, was carried in the genetic code to the population, creating quite a few of the individuals who could trace their 5% of the island's residents could trace their heritage back to that one survivor who had that gene. So, serine was replaced with phenylalanine. That's one of your questions. So now it's your turn, okay? So take a look at these two circles. And what do you see? Can you see a number? Here's the results. If you have normal vision, if you see a 25 here and a 29 here, but you possibly, you need a doctor to tell you, a visual specialist to tell you for sure, if you see 25 here, but you just see spots here and you don't see 29. Okay, here's some more. You should see numbers in both of these circles. Normal vision you'll see a 45 there and a 56 here. But a colorblind person would just see spots here and 56 there. And then you should see numbers here as well. So take a look at those. Normal vision will see number 6. Normal vision will see number 8, but colorblind will see spots in both of those. And if you're colorblind, no, it does not mean that you are related to that man from that island, okay? Other people in the world carry the gene as well. It's actually quite popular. Okay, what number do you see here? I think this is the last slide. If you have normal vision, you will see the number 5. If you may have a form of color blindness called red-green color blindness, if you see the number 2 here. Pretty interesting. So that's our notes on DNA replication.